Welcome to 18.1, Part 1. We are talking about the Trp Operon. We're going to break up Chapter 18 into 3 sections so that we can wrap our head around it. So 18.2 will be the lack operon, and 18.1 Part 2 is going to be the lack operon, and 18.1 Part 3 is going to be positive and negative control. So this is all of bacterial gene expression. We're going to be talking about E. coli. So remember that this is bacterial. 18.2, we're going to start talking about eukaryotic gene expression. So let's keep in mind what it is that we are talking about. So every cell in our body contains the entire genome, the same genome. The cells in your brain that are brain cells have the same genome as the cells in your skin. And the difference is that the cells in your brain have the brain cell gene turned on, whereas the brain cell has the skin cell gene turned off. And so we call this gene regulation, when it is the turning off and on of our genes. So it is determining whether they're going to be expressed or not. And this is going to happen even at a smaller level or a shorter level of time. So even as your day progresses, our environment's going to change, and we need to be able to adapt to that. And we're going to be doing that continuously by using this gene expression. And let's look at an example of this. Let's look at, like we said, E. coli. That E. coli is a bacteria that lives in our intestines. And if you think about their environment, they're going to go through long periods of time where we're not eating, and so we're not providing it with tryptophan. And so it's going to need to make tryptophan when it's not around. And then when we are eating, there's going to be plenty of tryptophan so that it is not going to need to make it. Let's go back in time for a moment and remember what tryptophan is. Tryptophan is an amino acid. And our E. coli is going to need all of the different amino acids in order to make proteins. And so if those amino acids are present, then they don't need to make it. And if they're not present, then they're going to need to synthesize it. And so when an E. coli is not getting the tryptophan, it's going to go through regulation. So there are 2 different levels of gene regulation that we're going to talk about. So one is when we stop the cell from producing enzymes completely. And so we call this gene regulation because we are regulating the expression of our genes by at the genetic level. So at the gene level, we're going to stop the entire production of an enzyme. And so when we are talking about like more short time change or adaptation, we are going to, our genes are going to be expressed or not expressed due to feedback inhibition. And so this is just an immediate short-term adaptation where it's going to be coenzymes that are going to attach onto our enzymes to stop them from working. And that's going to just turn the enzymes on and turn the enzymes off. We're not actually stopping the production of them. We're just turning them on and off for a shorter period of time or for a quicker response. So E. coli synthesizes tryptophan in a 3-step pathway. So that means that it takes 3 enzymes. We have to make 3, or the E. coli has to make 3 enzymes in order to produce the tryptophan. And it's going to do this with 5 different genes. So it's going to be 5 genes that produce 5 different polypeptides that are going to make 3 different enzymes. There's going to be, right here, you're going to see the long strand of mRNA, and it's going to be stop and start codons here that are going to break up those the polypeptides during translation, and it's going to show it's going to indicate where translation should start and end, and this is going to separate the polypeptides. So the grouping of these genes together, these 5 genes in this case, in other cases it's a different number of genes, but we call this coordinately controlled, and that should make a lot of sense because it's coordinating the when the enzymes are going to be made so that they're made all together, and that's going to make the process more efficient. Instead of having the genes in different parts of the chromosome, it's going to make it so that this can just be a more efficient process. right here, if these are our 5 genes for the 3 enzymes. Right before it, we can see here's the operator. The operator is the on-off switch, and that's going to essentially turn on or off the expression of these genes. And it does it really simply. Here's the RNA polymerase. If there's a repressor molecule or a protein that's on the operator, it's going to block the RNA polymerase from moving downstream. And it's really that simple. So once you start getting confused and sort of overwhelmed with information, just go back to this, that it's just a repressor molecule that is going to be attaching onto the operator, and it's just going to be blocking the RNA polymerase from moving downstream. If it's blocked, it can't express it. And if it's not blocked, it's going to move downstream. So we have an operon. We call this an operon. The operon consists of the promoter region, the operator, and the operator can be in the promoter region or between the promoter region and the series of genes here. And so in these genes, so these 3 things, the promoter region, the operator, and the coding sequence, these are going to make up an operon. And this is the tryptophan operon, or for short, the Trp Operon. So the Trp Operon is turned on. By itself, it's just going to be running. It's just going to be left to make tryptophan because the gene is, because the cell is going to need it. The E. coli is going to need tryptophan. So if the polymerase can access it, it's going to transcribe it. So it's going to be turned off when there's a repressor protein on there. So we said that there's going to be a repressor protein right here, and it's going to be attached, and that's going to turn it off. It's going to block the attachment of RNA polymerase, and it can't be transcribed. So repressor proteins are specific to the genes that they inhibit. So the Trp repressor protein inhibits only the Trp operon. It's only going to stop or block the Trp operon. The repressor protein is coded in a regulatory gene. So let's wrap our heads around this picture. We have our RNA polymerase. We have our operator region here with our repressor protein. And we have our coding sequence further down. So if we go upstream, we're going to see that there is a regulatory gene right up here. This regulatory gene has its own promoter region. And so these regulatory proteins are going to be expressed continuously and at a slow rate. And they're going to be also reversible. So here, when this repressor protein is synthesized from the regulatory gene, then it's going to be in a off level, or not an off level. It's going to be translated in a way that means it's off. And in order for it to turn on, it's going to need a co-repressor. So let's say that again. So this protein, when it is translated, it's going to be translated in an off. And in order for it to work, it's going to actually require, and this should make a lot of sense, tryptophan is the co-repressor. So when there's tryptophan present, it's going to bind to we can see this picture here. When tryptophan is present, the co-repressor is present. It's going to bind to the protein. That's going to turn the repressor on. It's going to change its shape. Oftentimes these repressor proteins are allosteric. It's going to change its shape. And when its shape is changed, it's going to attach on and it's going to block the RNA polymerase. This should make sense because if tryptophan is present, then it doesn't need to make any more. So if tryptophan is already present, it's going to turn the repressor on, and the repressor is going to bind to the operator, and it's going to block the RNA polymerase. So that is going to turn off the operon, and the gene is no longer being expressed. Like we said before, the tryptophan acts as a co-repressor. So here's our repressor protein and our co-repressor. So one more time through, as tryptophan builds up, it will bind to the repressor and turn on the repressor, but turn off the gene or turn off the operon. When tryptophan levels drop off, So when this molecule here is not around, it's going to not bind to the repressor, and then the repressor is going to change shape and no longer bind to the operator. And that's going to allow RNA polymerase to zip down and make tryptophan. So this is a prime example of homeostasis. Remember that homeostasis is maintaining a stable environment when there are changes to the external environment. And so when we have tryptophan available, or when the E. coli has tryptophan available, it doesn't need to make it, and it does when it isn't available. And so this is homeostasis. Finally, here's our last slide. The Trp operon is referred to as a repressible operon because its default is set to on. So it's going to just be making, it's just going to be working unless it's being repressed, unless it doesn't need to make it. And so it's not going to make it's not going to express the gene if the tryptophan is present or if the substrate is present. So it's generally turned on unless the tryptophan is present. Anabolic reactions tend to be regulated by repressible operons. This is because they don't need to build new molecules if one is already made. If it's already there, we don't need to build it. So this is going to be different for inducible operons, which we'll talk about next. I hope this helps, and let me know if you have any questions.